Hello students, it's 3 p.m. It's time to start the class. But let's start 5 minutes later when more people would join this class. So last class I didn't see many people, but I have uploaded the recorded lecture. For some reason my screen is still visible from only a corner. I have made some amendments today. Let us see how does it turn out. But it's always better to attend the live classes. So let's start at 3:05 p.m., allowing 5 minutes more for students to join this call. Meanwhile, everyone who requests to join this call will be allowed to this class. So it's 3:05 now, 5 p.m. We will start the class. So hi everyone, my name is Ashutosh Behera and I am the teaching assistant for this course, Basics in Biology. And we are in here together for solving practice problem set assignment for week 6. So all those lectures and classes and topics covered in lecture week 6 will be uh, will be shown today. Problems from such lectures which are exemplar assignment questions which will help you understand the concepts better as well as solve your assignments and prepare you for the quiz, right? So general set of instructions before starting the presentation and starting the class. So this is a question and problem solving session. So I will post 10 to 15 questions from the lecture week 6, since we are doing lecture week 6 today, and then we will try to solve multiple choice option questions. We will try to solve the correct answer, and then I will brief you about the concept and rationale behind the correct answer so that the concept in relation to the correct answer is also clear. And this is a session which is happening on Saturdays 3 p.m. to 5 p.m. and if we finish early or later, that's depending on the questions and the kind of response we get in the class. And this will be an interactive session, but I would request all the participants to please turn off your video chat and your mic. You can interact with me via the chat box, ask your questions, keep your answers ready when I display the question on the screen. If you have any doubts, if you want to talk to me, you can do so through the chat box. I will be following the chat box closely. And okay, with that much of an introduction, let me present my slides. I will share my screen. My screen is set. Open my presentation. Just a minute. Okay, I hope everyone can see my screen and follow. Can you see the slides changing? I hope it's visible to everyone. Please let me know in the chat box with a like or a yes. Very good, very good. Okay, so we are going to solve practice problem set for week 6 today. And as I told you earlier as well, my name is Ashutosh Behera and I'm a PhD scholar at IIT Kanpur. The instructor for this course, Basics of Biology, is Professor Vishad Trivedi from IIT Guwahati. Okay, the way we proceed here is I will display the question on the screen and read out the question out loud with all the options, and you guys have to tell me what is the correct answer. Even those who will be watching this video, this is the same exercise we will be following with the in live participants as well as who will watch the recorded video. Okay, without any further ado, let us move to the first question of this session. Question number 1. In central dogma of life, the enzyme used for transcribing the genetic information is dashed. This is the question. In central dogma of life, enzyme used for transcribing the genetic information is option A, RNA dependent RNA polymerase. Option B, RNA dependent DNA polymerase. Option C, DNA dependent RNA polymerase, or option D, DNA dependent DNA polymerase. Quite a few options, confusing options, but only one of them is correct. One of them is a key enzyme used for transcribing the genetic information in the central dogma of life. Let us wait for a minute to collect some answers. Think about it, read the question carefully, and tell me what you think is the correct answer. Okay, time is up. Let us see what are the answers that we have received. Sahil has answered option C, and I don't see any more answers. Aparna and Aishwarya, maybe they are having. Okay, Aishwarya has answered option C. Okay, let us see what is the correct answer to this question. Sahil and Aishwarya are both right. Correct answer is option C, DNA dependent RNA polymerase. So, DNA dependent RNA polymerase is the enzyme that is important for transcription of genetic information in the central dogma. So, now what is this enzyme? The enzyme responsible for transcribing genetic information from DNA to RNA. So, in the central dogma, the first step in a central dogma is copying of information from DNA to RNA. DNA is usually double stranded and it's consisting of deoxyribonucleic acid, while RNA may be double stranded or single stranded, but usually is consisting of ribonucleic acid. So the transfer of information from a DNA to an RNA is carried out by this enzyme through a process called transcription, which is in short known as RNA polymerase or DNA dependent RNA polymerase. Now in prokaryotes like bacteria and archaea, a single RNA polymerase transcribes all type of RNA. So there are many types of RNA found in organisms. mRNA stands for messenger RNA, tRNA is the transfer RNA, rRNA is the ribosomal RNA. These are all different RNA molecules playing very distinct functions. mRNA or the messenger RNA is the one which will further carry the genetic information, translate the genetic information into proteins. tRNA is involved in translation of the mRNA, and rRNA also forms an integral part of the ribosomal subunits. Now in prokaryotes there is a single RNA polymerase that transcribes all types of RNA. But what happens in eukaryotes? In eukaryotes there are three main RNA polymerases. RNA polymerase 1, which transcribes rRNA except 5S rRNA. RNA polymerase 2. Polymerase 2 is the most important enzyme because it transcribes mRNA or the coding genes. Genes which are present in the DNA are transcribed into mRNA by RNA polymerase 2 and some small nuclear RNA or snRNA. And RNA polymerase 3 transcribes tRNA, 5S rRNA and some other small RNAs. Here is a representative diagram showing the work of DNA dependent RNA polymerase. DNA dependent RNA polymerase undergoes a process, carries out a process called transcription, which is as shown here, melting of the double helical structure of the double stranded DNA, and then synthesis of a nascent RNA or messenger RNA using DNA as a template. That is why this enzyme is also known as DNA dependent RNA polymerase. So this is the coding strand, this is the template strand. So the DNA strand with a polarity from 3' to 5' is taken as a template and complementary to which the mRNA is made by this enzyme DNA dependent RNA polymerase. Now, looking at the different kinds of RNA polymerase we have in eukaryotes, RNA polymerase 1 or Pol1 transcribes RNA except 5S rRNA. It is located in the nucleolus, produces 28S, 18S, and 5.8S rRNA important for ribosome formation. So, all these rRNA are integral part of the ribosomes. And by S, when I mean S is standing for sedimentation constant. RNA polymerase 2 transcribes mRNA located in the nucleoplasm or inside the nucleus, produces mRNA, small nuclear RNA, snRNA, miRNA or microRNA involved in gene expression and regulation. So Pol2 is the major enzyme. Then comes Pol3, RNA polymerase 3 transcribes transfer RNA or tRNA and 5S rRNA. It is also located inside the nucleus, produces tRNA, 5S rRNA and some small RNAs important for translation and RNA processing. So these different enzymes have distinct functions, but in prokaryote everything is carried out by a single DNA dependent RNA polymerase. In eukaryotes, if you just talk about gene expression and regulation, then major function is carried out by DNA dependent RNA polymerase 2. So the correct answer is, as the students have mentioned, option C, DNA dependent RNA polymerase is involved in central dogma of life for transcribing genetic information. Okay, is it clear to the students? We touched up a bit upon all different kinds of polymerases and what their function is, so that you always remember which is for which. Okay, great. Moving on to the second question. Which of the following combination of events in the central dogma of life are possible? And there is also a hint here, do not consider in vitro experiments. So there is no in vitro experiment, but in natural settings with organisms, which of the following combination of events in the central dogma of life are possible? Option A, DNA to DNA, DNA to RNA, RNA to protein, RNA to DNA. Option B, DNA to DNA, DNA to protein, RNA to protein, RNA to DNA. Option C, DNA to DNA, RNA to protein, RNA to DNA, protein to DNA. Option D, RNA to DNA, RNA to protein, RNA to DNA, protein to RNA. Look at this question carefully, go through all the options, and only one of them is correct, where all the mentioned combination of events are possible in central dogma of life. Let us wait for a minute and collect some answers. I will reveal the correct answer in a minute or so. Come on, since we are a thin class, I would expect all of the students to answer this question. It's okay if you do not know the correct answer. Even if your answer is wrong, do not need to feel bad. We will understand about the correct answer and you will not make the same mistake again. So I urge everyone to participate, as well as those watching this video. Okay, the time's up, and let us see what is the correct answer. Let me first check for answers in the chat box. Okay, Aishwarya says option A, and Sahil is saying option B. Okay, so let me reveal what is the correct answer. The correct answer is option A. Congratulations, Aishwarya is correct. Option A is the correct answer to this question. DNA to DNA, DNA to RNA, RNA to protein, RNA to DNA is the correct combination of events that is naturally possible in central dogma of life. Now let us first know what is this central dogma we keep talking about. Central dogma says there is one way flow of genetic information from DNA to RNA to protein. The central dogma of molecular biology is a theory stating that genetic information flows only in one direction from DNA to RNA to protein. So this was the initial central dogma. Then what happened? People discovered retroviruses, which are organisms in which there is a conversion of RNA to DNA and back from DNA to RNA. So then the central dogma got modified. The genetic information flow happens from DNA to RNA to protein, and DNA and RNA are interchangeable. RNA can directly go to protein, or RNA can go to DNA and then follow the central dogma again. So the DNA can form more DNA by process called DNA replication. It can transcribe to RNA, double-stranded DNA transcribing to a single-stranded RNA or messenger RNA. Then this messenger RNA can then be translated into functional units called protein. Now in retroviruses, which are special cases, RNA can also go back to DNA through a process called reverse transcription. And this is the whole central dogma of life. Now let us evaluate each option that we have seen in the question. So, for your reference, central dogma, it's a natural process. DNA to DNA is replication, as I said. DNA to RNA is transcription, which we encountered in the first question. It is carried out by polymerase 2. RNA to protein is called translation. Translation of mRNA to polypeptide is carried out by ribosomes, which are protein synthesis factories of our cells. RNA to DNA happens in reverse transcription seen in retroviruses, as I was mentioning. Protein to DNA cannot happen, not naturally possible. Protein to RNA is also not naturally possible. So the one-way flow of genetic information cannot be reversed from protein back to nucleic acids. So in option A, we have DNA to DNA, which is replication, DNA to RNA, which is transcription, RNA to protein, which is translation, and RNA to DNA, which is reverse transcription. And option B is not correct because DNA to DNA replication is correct, but DNA to protein cannot happen directly. This does not occur directly, mRNA is needed. So direct conversion of DNA to protein cannot happen in natural settings. While the other two options are correct, RNA to protein is translation and RNA to DNA is reverse transcription. While in option C we have protein to DNA, and in option D we have protein to RNA, which is not naturally possible. That is why option B is also a wrong answer. But option A is the correct answer, where we have replication, DNA replication, where we have transcription of DNA into mRNA, translation of mRNA into protein, and reverse transcription of RNA to DNA. So the correct answer to this question is option A, which follows all the natural processes or combination which you can find in central dogma. Is it clear to the students? Let me know in the chat box and we will move forward. Great. Thank you. Okay. Moving on to the third question. Taq DNA polymerase was found from Thermus aquaticus in Dash Hot Springs in Yellowstone National Park that works at an optimum temperature of Dash with Dash as cofactor. So this is a fill in the blanks question and you have 3 fill in the blanks. So 3 answered options. Option A, 276 degree Fahrenheit, 72 degrees Celsius, magnesium ion. So I'm reading the answers in the order of the options. Thermus aquaticus in dash hot springs. The temperature of the hot spring is the first answer, 276 degree Fahrenheit. The next dash is 72 degrees Celsius and magnesium ion, and thereby we have four options. Option B, 276 degree Fahrenheit, 92 degrees Celsius, cobalt ion. Option C, 176 degree Fahrenheit, 72 degrees Celsius, cobalt ion. And option D, 176 degree Fahrenheit, 72 degrees Celsius, and magnesium ion. Note that for the option to be correct, all three of the fill in the blanks should be correct. Taq DNA polymerase was found from Thermus aquaticus in dash hot springs. What is the temperature of the hot spring in Yellowstone National Park? And the enzyme works at an optimum temperature of dash, and it needs dash as a cofactor. Let us wait for a minute and collect some answers. I will reveal the correct answer in a minute or so, and then we will go with the brief explanation. This is purely a memory based question, but this is an important discovery and you guys should remember this. This will help you a lot. Which option do you think is the correct answer? I hope you are using your own mind and not Googling it. Do not rely on Google. Remembering it by yourself and following the lectures will help you in the long run. Sorry. Okay, that's enough time and time for me to reveal what is the correct answer. So the history of discovery of the Taq polymerase and what is the correct answer? The correct answer is option D, 176 degree Fahrenheit was the temperature of the hot spring in Yellowstone National Park, but the enzyme works at an optimum temperature of 72 degrees Celsius with magnesium ion as the cofactor. So Aishwarya has again answered correctly, and it's okay, Sahil, if your answer is wrong. You do not need to feel shy. Keep participating. I like the response. Okay, so this is a picture of a geyser or hot spring, which we call at Yellowstone National Park, and the yellow color that you see is due to which the national park is called Yellowstone National Park. The yellow color is from the sulfur deposits in the hot spring. So Taq polymerase was discovered in this bacterium Thermus aquaticus, that was mentioned in the question as well. The hot spring where Thermus aquaticus was discovered in Yellowstone National Park had a temperature of around 158 to 176 degree Fahrenheit, which is roughly 70 to 80 degrees Celsius. So imagine in degrees Celsius, the bacteria is thriving at high temperatures of 70 to 80 degrees Celsius. This was discovered by Thomas D. Brock and Hudson Fries in the year 1969. And what is its significance? Taq polymerase, since it is isolated from the thermostable bacteria, Thermus aquaticus, Taq polymerase, the enzyme is heat stable, making it essential for polymerase chain reaction. So all the modern day PCRs run with Taq polymerase. In 1985, Carey Millis used Taq polymerase to revolutionize PCR, leading to rapid DNA amplification. So PCR is rigorously and very regularly used in laboratories to amplify our DNA using this enzyme, which is heat stable Taq polymerase. This enzyme withstands high temperatures of 95 degrees Celsius needed for DNA denaturation, unlike regular DNA polymerases that denature at high heat. So even at 95 degrees Celsius, many other polymerases denature, while this Taq polymerase stays intact. Now, what are the optimal conditions for Taq polymerase to work? Optimal temperature is 72 degrees Celsius or 162 degree Fahrenheit for DNA synthesis. While Taq polymerase remains active at high temperature, it works best at 72 degrees Celsius, the extension step in PCR. So in PCR we have the initial denaturation, the annealing and the extension. So in extension step, the temperature is always set at 72 degrees Celsius because Taq polymerase has an optimum at 72 degrees Celsius. It can withstand denaturation at 95 degrees Celsius without losing function. And what is the cofactor requirement? Cofactor requirement is the Mg2 plus or magnesium 2 plus ions act as a cofactor for polymerase, essential for catalytic activity and stabilizing the DNA polymerase complex. So the DNA polymerase needs to access the DNA, bind to the DNA, and then undergo polymerization or replication. So that can only happen when the complex is stabilized with the help of magnesium 2 plus ions, typically provided as MgCl2 in PCR reactions. So in the buffer that you use in PCR, you have high concentration of MgCl2, which provides the required Mg2 plus ion for proper functioning of this RNA polymerase 2 complex. So if you can see the structure of this protein RNA polymerase 2, which is already inbound to a DNA molecule, the green here is the bridge helix. The DNA and the bridge helix is coordinated by a coordination bonds from Mg2 plus ions. So thus the bacteria that was isolated from a hot spring of 170 degree Fahrenheit, optimal working temperature of 72 degrees Celsius, and cofactor requirement of magnesium 2 plus ion for Taq polymerase. I hope it's clear to everyone. If yes, then we can move to the next question. Very good. Very good. Thank you. Moving on to the fourth question. What is the antisense strand for the mRNA sequence? 5 prime UAC GGAU to 3 prime. So this is the orientation from 5 prime to 3 prime, and you have to tell me what is the antisense strand for this mRNA, where U stands for uracil, A is adenine, G is guanosine, and C is cytosine. What is the antisense strand for the mRNA sequence? Option A, 3 prime ATGCC TACG 5 prime. Option B, 5 prime TACGG ATGC 3 prime. Option C, 3 prime TACGG ATGC 5 prime. Or option D, 5 prime ATGCC TACG 3 prime. So there are basically two sequences in the options which are there in different orientation. So you have to first understand what is the antisense strand, and then you might require a pen and paper for this. What is the antisense strand for this mRNA? Let me wait for a minute to collect some answers, and then I will reveal the correct answer. Okay, I hope you guys have worked out for the antisense strand of this mRNA. Let me check the chat box for some answers. Okay, I don't see any answers. I think this was a little challenging for people. Oh wait, Sahil has answered. Sahil is answering option C. Okay, thanks for the answer, Sahil. Let me reveal the correct answer to this question. Option A is the correct answer to this question. Option A 3 prime, ATGCC, TACG 5 prime is the correct answer. Sorry Sahil, this time you were not correct with the answer. Option A is the correct answer. So let us understand why is it option A. So let us first understand what is a sense and what is the antisense strand. So in a gene or wherever there is transcription going on, what is the sense and antisense strand? The antisense strand is also the strand which starts with 3 prime to 5 prime. The sense strand is always the one which is from 5 prime to 3 prime. So here as well in the question we have 5 prime to 3 prime as the sense strand. And the antisense strand will always be opposite of that from with the polarity at least from 3 prime to 5 prime. So that rules out two options, option B and D. Option B and D cannot be the correct answer since they have the similar orientation to the sense strand. So it can be either option A or option C. So this is the sense and antisense strand. When mRNA sense transcript is made, it is made in the 5 prime to 3 prime direction since the antisense strand is taken as a template. Right? Now let us come to this answer. To determine the antisense strand for this given mRNA sequence, we have to follow these steps. How? The antisense strand, also called the template strand, which is the strand from 3 prime to 5 prime, is complementary to the mRNA sequence and is written in DNA basis. So one thing it is clear, the sequences are complementary to the mRNA and it is written in DNA basis. And when I say DNA basis, you replace all the U. Since mRNA uses uracil instead of thymine, we need to replace all the U with T when determining the DNA strand. The base pairing rules, these are the base pairing rules for common knowledge. Adenine pairs with T, we have A to T bond, U to A bond, C to G bond and G to C bond. Adenine with thymine, uracil with adenine, cytosine with guanine and guanine with cytosine. So writing the complementary DNA strand for this sequence, we get for U the complementary DNA strand is A, for A it is T, for C it is G, for G it's C again, G C, A T, U A, G C, C to G. This gives the sequence 3 prime, T G C C T A C G 5 prime, which matches option A. Sorry, A T G C C T A C G. A T G C C T A C G, which is option A. So the correct answer is option A, 3 prime to 5 prime, A T G C C T A C G. I hope it's clear to the students. Let me know in the chat box with a like or a yes or a... Very good, very good. Likewise, keep participating. We will reach the correct answer. We will understand how the correct answer has been reached, or what is the step you need to follow to find out or to tackle such questions in the future. Okay, for all those who have joined recently and who are new to this class, a common set of instructions. Please turn off your video chat and mic during this session. For smooth conduction of this session, you can interact with me via the chat box. I will be monitoring the chat box constantly, and this is a question answer session in which I will display the question on my screen, and there are 4 multiple choice options out of which 1 is correct. So you guys have to let me know in the chat box what do you think is the correct answer. Okay, moving on to the next question, question number 5. What is the mRNA sequence if the DNA sequence is 5 prime, ATG, CCG, TAT, TGC, TAG, CC, 3 prime is a sense strand? So this question is kind of related to what we had in the previous question. Here the DNA sequence is given and now you have to find out the mRNA sequence. and DNA sequence is given in the sense strand. So please note everything. You might require a pen and paper to solve this. I will read out the options. Option A, 5 prime CCGATC GTT 3 prime. Option B, 3 prime AUG 5 prime. Option C, 5 prime AUG 3 prime, and option D, 3 prime CCGA 5 prime. So these options are also very similar with two different sequences in 5 prime to 3 prime direction and one of these options from 3 prime to 5 prime direction. So now take all of your time, read the question carefully, what is given and what is asked, and then let me know what you think is the correct answer to this question. If the DNA sequence in the sense strand is given, how to know what is the mRNA sequence? Let me wait for a minute or more than that, because this answer might take some time, and let me know what do you think is the correct option. At the end of the minute, I will reveal my correct answer. Excuse me for a minute, I'll drink some water and come back. I'll be right back since you have been solving this problem. Okay, that's a lot of time now. Let me check the chat box for some answers. I see Aishwarya has answered option C, and there are no more answers. Okay, I applaud for your confidence, Aishwarya. Let us see what is the correct answer to this question. What is the mRNA sequence if the DNA sequence is given in the sense strand? And the correct option is option C. Well done, Aishwarya. You have calculated it correctly. Option C is the right answer. 5 prime to 3 prime. AUG, CCG, UAT, TGC, UAG, CC. Now let us understand. So again, the sense and antisense strand. The sense strands code for genes. Now which is the sense strand? The sense strand is this, the 3 prime to 5 prime. Sorry, it's reversed here. The antisense strand used as RNA template. Okay, antisense strand is always the 3 prime to 5 prime one, which is now used as the template for transcribing RNA, and the sense strand is always coding for genes. Now the RNA that will come as a complementary sequence to the antisense strand of the DNA. RNA that encodes for the gene comes from the antisense strand, and uh, three of these form a codon, which is now translated into amino acid language. Okay, so for this question, to determine the mRNA sequence from the given sense strand, we will follow these steps. Understand the sense strand. The sense strand, coding strand of the DNA, has the same sequence as the mRNA, except that thymine in DNA is replaced by uracil in mRNA. Why is it so? Because if you remember, sense strand is a top strand, which is 5 prime to 3 prime. Now we already have an antisense strand which is complementary to the sense strand, right? And then the mRNA, which strand of both these strands will be used as a template for mRNA? It is the antisense strand, which is already complementary to the sense strand. So the sense strand and mRNA will share sequence similarity. Only difference in the sense strand of the DNA and the mRNA will be uracil and thymine, because in mRNA we have uracil instead of thymine. So in the transcription process, DNA 5 prime to 3 prime ATCG, ATCCGG, TAT, TGC, TAGCC will be transcribed into mRNA 5 prime AUG. So wherever there is T, we just replace it with U here, two more U's here, and a U here, replacing T with U, and the correct answer is therefore option C, 5 prime AUG, CCG, GUA, UUG, CUA, GCC 3 prime. So whenever you have sense strand of the DNA given, you just copy it into the RNA and change the thymine to the U basis, then you get your mRNA. Now whenever there is antisense strand given, you have to find complementary bases to now find the sequence of the mRNA. Is it clear to the students? This might, if it is difficult for you guys to understand, I will explain it again. Oh great, you guys understood it in the first time. Very good. Cool. Moving on to the next question, question number 6. Question number 6 is a statement based true and false question. What is the statement? In intrinsic mechanism of termination in transcription, rho factor is not essential. The presence of GC rich hairpin followed by 7 U residues is required. This is the first statement, or statement A, assertion, and then the reason is the formation of hairpin structures which have weak RNA DNA hybrid interactions signals the dissociation of the polymerase and the transcript. Understood? These are the both statements. One is the assertion and one is reason. Now there are these options. Both A and R are true, and R is not the correct explanation of A. Option B, both A and R are true, R is the correct explanation of A. And option C, A is true but R is false. And option D, A is false but R is true. Understood the question? I will repeat the question for you again. There are two statements, assertion and reason. The assertion says, in intrinsic mechanism of termination, rho factor is not essential. You only require GC rich hairpin followed by 7 U residues. And then the reason is because formation of hairpin structures which have weak RNA DNA hybrid signals the dissociation of the polymerase and the transcript. So you have an assertion, you have a reason. Now you have to say whether A is correct, whether R is correct, whether A and R are both correct, and R is the explanation of A or not. Right? I think it is easy to understand, but read through the question carefully and take your time to answer this. I will wait for a minute, check the comment box for answers, and then reveal the correct answer to this question. Okay, I think it's enough time. A minute has already passed. And let me check the chat box for some answers. Do we have any answers? Oops, we do not have any answers. I think people are having a hard time dealing with lecture week 6, or they are not, maybe they are not well read, or they are having difficulty in learning the concepts of transcription. It's fine, you do not have to worry about it. I will cover as much as possible through these questions of what you need in your quizzes and for basic understanding. Okay, if there are no answers but still I'll reveal what is the correct answer to this question. Correct answer is option B. Both the assertion and the reason are true, and the reason is the correct explanation of assertion. Right, so B is the correct answer to this option B to question number 6. Now let us understand how does termination of the DNA transcription happen. So there are basically two methods of termination of transcription. One is the intrinsic termination and a row dependent termination. Now what is a row dependent termination? In a row dependent termination there are certain sites in the DNA which are called row binding sites, and these sites are bound by the row proteins, row protein mediated termination. And when the row protein is bound, which signals the polymerase to stop. Like polymerase, you have to stop here, you do not transcribe any further because row protein is sitting on the row binding site and is signaling the polymerase to do so. But what happens in intrinsic termination? It does not require any row protein. Rather, it depends on the formation of such hairpin structures where there is a stretch of repeat nucleotides, which is a GC rich stem and a poly UUUU mRNA. So this confers intrinsic termination signal and is responsible for row independent DNA transcription termination. So we learnt about two different types of termination. Now what happens in intrinsic termination? In intrinsic termination there are these repeats which are called inverted repeats. You can see here CAG TCGA. So inverted repeats will have the same sequence here in the inverse fashion, CAG TCGA. Now what happens when RNA is formed from such invert repeats? So these invert repeats are marked by poly A signals or poly A stretch of adenine nucleotides, which turn into poly U when they are transcribed into RNA. Now these form complementarity, these invert repeats form complementarity. The pink has complementarity to the pink, and the green forms a hairpin loop structure. This kind of a structure is called hairpin loop structure. The presence of RNA hairpin caused by the inverted repeats or on a GC rich sequence followed immediately by U rich region downstream will cause the termination of transcription, because the DNA RNA hybrid is the weakest when there is uracil. And why is it so? Because if you remember, adenine to thymine or the pyrimidine interaction. So adenine to thymine interaction, adenine to thymine and adenine to uracil interaction is a double bonded interaction. It is connected by two hydrogen bonds. While GC interaction or cytosine guanine interaction is a three hydrogen bonded, is a very strong interaction. So AU, multiple AU such interaction, multiple weak interactions cause the RNA polymerase to fall off, cause the separation of the mRNA transcript from the DNA template. This is the process of intrinsic termination, with the formation of these hairpin loop structures followed by a poly U stretch. Poly U weakens the DNA RNA hybrid due to its dual hydrogen bond instead of triple hydrogen bond of the GC. And when many such repeats are present, then the mRNA falls off from the DNA due to weak interactions. Okay, is it clear to the students? I will ask again. Every time we learn about an explanation, I will ask to the students, and only when they are clear, we will move forward. Very good. Thanks for the confirmation. Moving on to the 7th question. Again, Seventh question is related to DNA transcription termination. Rho factor is an ATP dependent helicase which is required for termination of transcription. Rho protein is a dashed. Option A, dimer. Option B, trimer. Option C, tetramer. Option D, hexamer. The question is about the Rho protein. Now, what is the oligomeric state of the Rho protein, functional Rho protein? Is it a dimer, a trimer? Is it a tetramer or a hexamer? Let us wait for a minute and collect some answers. Okay, it's time for me to check the comment box for answers. And I see Sahil and Aishwarya have both answered option D. I'm happy to reveal that option D is the correct answer. Well done. Sahil is correct and Aishwarya is also correct. Rho protein is a hexameric protein in its functional state. Now let us see the structure of Rho protein. So this is the Rho protein. This is the amino terminal domain, contains the primary binding site, and the carboxy terminal domain contains the secondary site. When I say amino and carboxy terminal of the protein, amino terminal is considered the start of a protein and carboxy terminal is the end of a protein, because the protein is nothing but a long polypeptide chain folded onto itself, forming a 3D structure. So in a polypeptide chain, every polypeptide starts with the free amino terminal and ends with the free carboxyl terminal because of the peptide bond formation. Now, in this structure, we can see that Rho factor consists of 6 such subunits. And the first step happens when it recognizes these sequences called RUT sequences on the mRNA and on the DNA where the Rho factor binds, and then it reels in the DNA it reels in the mRNA leaving behind the polymerase and the DNA complex. Right? Now what happens in Rho dependent termination? So this is the RUT site that we saw in close detail, where the Rho factor is bound, which is a 6 oligomeric hexameric protein bound here. Now this will reel in or pull in the mRNA which is coming out of the transcription bubble. It will pull in, reel it back, and then separate the mRNA from the DNA. Now when the mRNA is separate from the DNA, RNA polymerase 2 will leave the transcription bubble complex, and thus we have the separate termination of transcription via Rho dependent mechanism. So in Rho dependent termination is a mechanism used in prokaryotes such as E. coli to stop transcription of RNA. It relies on Rho protein, which is an ATP dependent helicase. So helicase causes the reeling in of the mRNA, and it is ATP dependent. It means it is an energy expenditure process. Steps of Rho dependent termination: Rho protein binding. The Rho factor binds to specific RNA sequence called Rho utilization site or RUT site, which is rich in cytosine nucleotides and lacks secondary structure. It is pretty plain and elongated. It is enriched with C nucleotides, right? C. Translocation along the RNA. The Rho protein moves along the RNA in 5' to 3' direction using energy from ATP hydrolysis. Then what happens? Then the RNA polymerase pauses on the DNA. When RNA polymerase encounters a termination signal, often a GC-rich region that slows polymerase, it pauses. And then what happens in the final step is the disruption of the transcription complex. The Rho protein catches up with the paused RNA polymerase. Using its helicase activity, Rho unwinds the RNA DNA hybrid in the transcription bubble. This causes RNA polymerase to dissociate, releasing the RNA transcript. I hope it's clear to the students. You both have answered very correctly, but we also learned about all the detailed processes of Rho dependent termination of transcription in prokaryotes. I hope it's clear to the students. Let me know in the chat box. Very good. Okay, an hour has passed and we are past the 50% of the questions today. I have prepared 15 questions. Moving on to the question number 8. Which transcription factor recognizes the Tata box in eukaryotes? Now coming on to transcription factors. Which of these transcription factors recognizes the Tata box in eukaryotes? Option A, TF2A, option B, TF2B, option C, TF2C, or option D, TF2D. Come on guys, let us wait for a minute and then I'll reveal the correct answer. I will collect some answers from the chat box. Think about it. Which is the transcription factor that first recognizes the Tata box? Okay, I think that is enough time for this small question. Let me check the answers. Sahil has answered option D. Okay, and I don't see any more answers. And Sahil is right. Option D is the correct answer. TF2D is the transcription factor that recognizes Tata box in eukaryotes. Now to describe the assembly of the transcription machinery on the eukaryotic promoter, these are the events that happen. Now what is the Tata box? Tata box is nothing but the promoter region in eukaryotes. Why is it called the Tata box? Because it is enriched in sequences of TATA binding. Now what is the first event that happens? This green transcription factor called TF2D, transcription factor 2D, binds to the Tata box through TBP. Now what is TBP? Tata box binding protein. Now this is the part of one of the domains of the TF2D. Then comes TF2B and TF2A binding to the promoter in complex with TF2D. RNA pol to TF2F, TF2E, H bind to form the transcription initiation complex. So E and H are coming here. Then RNA polymerase is also recruited along with many other factors, now which forms the active transcriptional complex. What are the functions of D and H? TF2D contains TBP, the Tata binding protein. It binds the Tata box and marks the promoter as an active promoter for transcription. And what is the function of H? TF2H contains protein kinase that phosphorylates C terminus of RNA polymerase 2 so that it releases the promoter to begin elongation. So for C terminus tail phosphorylation of RNA polymerase 2 is important for transition of initiation into elongation where the actual mRNA is synthesized. And TF2H also contains DNA helicase that separates the DNA strands. So this is the common hierarchy of assembly of transcription factors at the promoter sites of eukaryotes. First come TF2D with its Tata binding protein binds to the Tata box, then recruits TF2B and A, and then which recruit E and H and polymerase and F to form the active complex. Then TF2H phosphorylates C terminal domain tails as well as unwinds the DNA for transition from initiation to elongation. So the correct answer is as Sahil mentioned, which is TF2D that binds to the Tata box as the first protein to bind to the Tata box in the promoter region. Is it clear to the students? I think, yeah. Is it clear to the students? If yes, then we can move to the next question. Okay, question number 9. In the post-transcriptional modification of mRNA, capping dashed, option A, starts as soon as 20-30 nucleotides are formed. Option B, starts along with mRNA synthesis. Option C, starts after the complete formation of mRNA molecule. Option D, does not happen. So here we are talking about post-transcriptional modification of mRNA, which is capping, one of the post-transcriptional modification. When does it start or does it not start at all? Does it start as soon as 30 nucleotides are formed? Does it start along with mRNA synthesis or after the complete mRNA is formed? Or it does not happen? Let us wait for a minute to collect some answers and then I will reveal the correct answer. Okay, it's time for me to check the chat box and reveal the correct answer. Let me see. Aishwarya has answered, I think, option A. Okay. And there is no other answer in the chat box. Okay, let me reveal what is the correct answer. Correct answer is option A, starts as soon as 20-30 nucleotides are formed. Well done, Aishwarya, your answer is correct. Starts as soon as 20-30 nucleotides of mRNA is formed, the capping starts. Let us learn in detail about mRNA capping. So this is the cap structure of, in the panel A we see cap is also called trimethyl guanine, 7-methyl guanosine, right, and trimethylation. And this is the mechanism for cap formation. There are many enzymes involved in mRNA capping, and as a result, in eukaryotic gene expression, we have, as soon as the mRNA starts for lengthening up to 20 to 30 nucleotides, these enzymes are at play, and then add the 7-methyl guanosine cap with triphosphate at the end of the mRNA, 5' end of the mRNA, which is called the 5' capping. Then splicing happens, and then we have 5' cap dependent translation. If the mRNA does lack a 5' methyl 7-methyl guanosine cap or a poly A tail, then it can undergo degradation. Now, what are the process of mRNA capping? First, what happens is the phosphatase action. The triphosphate at the 5' end of the nascent mRNA is converted to a diphosphate by removing one phosphate group. And guanyl transferase action, a GMP, guanosine monophosphate, is added to the diphosphate using a 5' triphosphate linkage, unlike the normal 3' to 5' phosphodiesterone. Methylation by methyl transferases, a methyl group is added to the N7 position of the guanine base, forming 7-methyl guanosine. Additional methylation, in some cases adjacent ribose sugars on the first or second nucleotide on mRNA are also methylated at 2' OH group, forming cap1 or cap2 structures. This is the mechanism of mRNA capping. So mRNA capping begins co-transcriptionally. So mRNA capping and transcription occur side by side, meaning it occurs during transcription rather than after it is completed. Specifically, capping takes place once the nascent mRNA is about 20 to 30 nucleotides long. And why does this capping start early? It protects the nascent mRNA from degradation by exonucleases, and it also helps RNA polymerase II transition from initiation to elongation. So we remember when the mRNA is 20 to 30 nucleotide long, and the capping happens simultaneously co-transcriptionally, it also helps in speeding up of polymerase II for elongation. And it also marks the mRNA for proper splicing and export from the nucleus. So the mRNA must be exported out of the nucleus into the cytoplasm, where the ribosomes are present for its translation. So a methylated 7-methyl guanosine 5' cap marks the mRNA for export, and it also calls for proper splicing of the mRNA. That is why capping starts early, and capping starts as early as 20 to 30 nucleotide longs when the nascent mRNA is. I hope it's clear to the students whether Aishwarya had answered this question correctly. If you have any difficulty understanding, you can ask me again. I will teach you again. Okay. Great response. Thank you for the response. Moving on to the 10th question. Poly A polymerase, which adds poly A tail at the 3' end of mRNA, is dashed. Now we are entering the post-transcriptional modification topic. Poly A polymerase, which adds poly A tail to the 3' end of mRNA, is option A, template independent DNA polymerase. Option B, template independent RNA polymerase. Option C, RNA dependent RNA polymerase. Or option D, RNA dependent DNA polymerase. You have to be careful about what is dependent and on which it is dependent or independent. Now let me know the answer of this question in a minute or so. I'll be back in a minute. Just write back. I think it's enough time for me to reveal the correct answer. Let me check the chat box. Sahil and Aishwarya have both answered option B, and you guys are absolutely correct. Option B is the right answer to this question. Template independent RNA polymerase. Poly A polymerase is a template independent RNA polymerase. Now let us learn in detail about poly A tail formation or polyadenylation in the eukaryotes. Now polyadenylation is the process of adding the poly A tail, a sequence of multiple adenine nucleotides as long as 200 adenine nucleotides to the 3' end of eukaryotic mRNA. So just in the previous question we saw a 5' post-transcriptional modification of 7-methionguanosine. Now we are seeing a 3' end modification, attachment of a polyadenosine nucleotide tail. This modification enhances mRNA stability, nuclear export and translation efficiency. So these are the function of the poly A tail. Steps of the poly A tail formation: cleavage of the pre-mRNA. RNA polymerase 2 transcribes past the polyadenylation signal sequence 5' AAUAAA. This is the polyadenylation signal sequence. When that is transcribed, located 10 to 30 nucleotides upstream of the cleavage site, cleavage factors are recruited like this. Here if you see in this diagram on my left, there are many cleavage factors that are recruited. CPSF, CSTF, CFs recognize this sequence and cut the mRNA downstream. Addition of the poly A tail, poly A polymerase or PAP adds 50 to 250 adenine residues to the 3' end, and poly A binding proteins bind to the tail to protect it from degradation. And then the poly A tail helps in mRNA export from the nucleus. It also interacts with the 5' cap to facilitate translation initiation. Right? So the poly A tail as well as the 5' cap are both required and important for translational initiation or the formation of the peptide bond initiation. This is the poly A tail formation. So this is the structure of the poly A polymerase. Poly A polymerase, also known as PAP, is the enzyme that adds poly A tail to the 3' end of the mRNA. Unlike most RNA polymerases, poly A polymerase does not require a template. It simply adds a stretch of adenine nucleotides to the mRNA. Now when you have non-specific sequence or oligo addition to an existing mRNA, then you do not need a template. The only job of poly-A tail polymerase is to add polyadenine nucleotides. Since it works directly on RNA and does not rely on DNA or RNA template, it is classified as a template independent RNA polymerase. I hope it is clear to you guys. Albeit you guys have answered very nicely. Both of you have answered the correct answer. Okay, moving on to the next question. Question number 11. Which of the following amino acids are coded only by one codon each? This is a tricky memory based question. You need to check the codon table to come up with the answer. Which of the following amino acids are coded only by one codon each? Option A, tryptophan and valine. Option B, leucine, isoleucine. Option C, tryptophan and methionine. Option D, methionine and leucine. Come on, let me wait for a minute and then I'll reveal the correct answer to this question. Amino acids which are coded only by one codon. No other codon can code this amino acid. Okay, it's a lot of time now. Let me check the comment box and then reveal the correct answer. Aishwarya is saying I think option C, and there are no more answers in the chat box. Okay, so those two amino acids which are only coded by one codon each are tryptophan and methionine. Correct answer, Aishwarya. Tryptophan and methionine are the amino acids which are only coded by one codon each. Methionine, because it's the starting codon, is the start codon, it's coded by AUG. No other codon can code for methionine. And tryptophan, we will learn in a bit that it is also coded by one single codon. Now, how can an amino acid be coded by many different codons? This rule is known as codon degeneracy. Refers to the redundancy in the genetic code where multiple codons code for the same amino acid. So unlike the case that was asked in the question, there are many amino acids like isoleucine, leucine, where multiple codons code for the same amino acid. This property, why did this happen? This property helps make the genetic code more error tolerant and minimizes the effect of mutations. So any mutation which is now changing the genetic code will not be able to translate it to a different amino acid due to degeneracy of codons. Key features of the codon degeneracy: multiple codons for one amino acid. There are 64 codons but only 20 amino acids, meaning some amino acids are encoded by more than one codon. Example, leucine, which is coded by UUA, UUG, CUU, CUC, CUA, CUG, 6 codons. 6 codons can code for the same amino acid leucine when present in the mRNA. Degeneracy usually at the third position. The first two nucleotides in a codon are highly conserved, but the third position, called the wobble position, can often vary without changing the amino acid. For example, serine, UCU, UCC, UCA, UCG, only the third base changes, and these codons can still code for degenerate serine amino acid. Now here am I showing a wheel, amino acid codon wheel, where we can combine these letters and we can see that So how to read this wheel? First, the inner circle is the first nucleotide, the middle circle is the second nucleotide, and this is the third nucleotide. So for alanine, there can be codons which can code for alanine: GCG, GCA, GCC, GCU. All of these can code for alanine. Now, if you see such amino acids which only have one such possibility, which is UG, AUG, A, then U and G, which is methionine, the start codon marked by this triangle, and tryptophan, which is W, UGG. So tryptophan is coded by UGG and methionine is coded by AUG. So these were the two correct options which are coded by only a single codon. Was it understandable? Was it easy to follow? Please let me know in the chat. If not, I can repeat any topic for you guys to understand it better. Okay, great. Thank you for the confirmation. Slowly moving on to the 12th question. Which of the following is not a property of genetic code? I'll read the question again. Which of the following is not a property of genetic code? Option A, some of the start codons are GUG and UUG. Option B, there is no comma in genetic code. Option C, the genetic code is degenerate. Option D, the code is overlapping. Now, which of the following is not a property of the genetic code? Please let me know in the chat box in a minute or so. I'll check the comment box and then reveal the correct answer. Okay, that's enough time, and let me check the comment box. Aishwarya is answering option D, and again there are no more answers. I would like to say that Aishwarya is exactly correct. Option D is the correct answer. Genetic code is not overlapping. So this is, the code is overlapping is not a property of genetic code. Now let us understand about the other options. The genetic code has several key properties, but it is not overlapping. Let's analyze each option. Option A, some of the start codons are GUG and UUG. This is true. The primary start codon is AUG methionine, but in some cases GUG and UUG can also serve as start codons. So cell does try to take AUG as a start codon, but sometimes GUG and UUG can also recruit methionine to start the translation. There is no comma in the genetic code. This is also true. The genetic code is continuous and non-punctuated. There are no gaps or commas between codons. All this nucleotide is continuous. The genetic code is degenerate. This is also true. Multiple codons can code for the same amino acid, making the code degenerate. But the code is not overlapping. The genetic code is non-overlapping, meaning that each codon, a set of 3 nucleotides, is read separately and does not share bases with the next codon. So when you have a codon it does not overlap with another codon, at least from the same gene. Three units of nucleotide make a single codon, which is consecutive to another unit of nucleotides for a single codon. The genetic code is non-overlapping, meaning that each codon is read separately and does not share bases with the next codon. For example, in the sequence AUG, GCU, CAA, the codons are read as AUG, GCU, CAA, not as AUGU, GCG, and then stopping as GCU. So here is what I was saying earlier. The genetic material is non-overlapping. The start codon can be AUG most of the times, but GUG and UUG are also observed. And then they are non-overlapping and comma free. They do not have stops or separation in between. They tend to transcribe everything with a single codon by codon manner. So option D was the correct answer, that in the genetic code, the code is always non-overlapping. I hope it's clear to the students. Very good. Moving on to the 13th question. tRNA or transfer RNA is usually of dash ribonucleotides in length. What is the length of tRNA in ribonucleotides? Option A 15 to 26, option B 25 to 26, option C 75 to 96, and option D 50 to 66. What is the length of transfer RNA in ribonucleotides? Just let me know the answer to the chat box. I will wait for a minute. Okay, it's enough time and I would like to check the chat box. Sahil and Aishwarya has answered option C. Let me reveal the correct answer. 75 to 96 ribonucleotides in length is the length of transfer RNA. Well done guys, you have answered the correct option. So let us have a closer look at tRNA. So this is the structure of the tRNA. It's a clover leaf structure. There is an acceptor stem here. There is 3' OH or amino acid attachment site. There is a T loop, a variable loop, T loop, variable loop, anticodon loop, and D loop. So these are the different parts. These are the different parts of the tRNA. Now, in average length of the tRNA in bacteria is 76 to 78 nucleotides. In eukaryotes like humans, 76 to 90 nucleotides. In archaea, it is 75 to 85 nucleotides. So this is the average length of the tRNA molecule when flattened. Is it clear to the students? I have listed here the average length of the tRNA molecule for prokaryotes, for eukaryotes, and for archaea. Very good. Very good. Coming to the 14th question, tRNA or transfer RNA is dash in 3 dimension. What is the structure of tRNA in 3 dimension? Option A, L-shaped structure. Option B, V-shaped structure. Option C, clover leaf structure. Option D, inverted clover leaf structure. Which of the following is a structure of tRNA in 3 dimensions? Let me wait for a minute and then reveal the answers. Okay, it's time for me to check the comment box and reveal the correct answer. Aishwarya has again answered option A, and there are no more answers. tRNA is actually an L-shaped structure in 3 dimension. Option A is the correct answer. Well done, Aishwarya. tRNA is an L-shaped structure in 3 dimension. So this is how a tRNA looks like. In a 2-dimensional system, tRNA looks like a clover leaf. The clover leaf which is used as a good luck charm. Right? This is an example of an alanine tRNA charged with alanine, amino acid binding site. There is an acceptor stem and the anticodon loop, the D loop as well as the T5 psi loop. tRNA 3D linkage, tRNA 3D landscape. The 3-dimensional structure of the tRNA is L-shaped, which is crucial for its formation in translation. This L shape allows tRNA to fit perfectly within the ribosome, bridging the mRNA codon and the amino acid it carries. So L shape is important for RNA function, for bridging the mRNA codon and amino acid it carries. So is it clear to the students? Aishwarya has answered correctly. L-shaped 3-dimensional structure of the tRNA. Very good. Moving on to the last question of this session, question number 15. Which of the following set of combination is correct regarding a tRNA molecule? Option A, 5' CCA end, T-arm contains pseudouridine, D loop often contain dihydrouridine. Option B, 3' CCA end, T arm contains pseudouridine and D often contains dihydrouridine. 5' CCA D loop contains pseudouridine. T arm contains dihydrouridine. Option D, 3' CCA and D loop contains pseudouridine and T arm often contains dihydrouridine. I will wait for a minute and then collect some answers and reveal the correct answer to this question. Regarding the tRNA structure, there are certain arms and loops and ends which are important in the tRNA structure. Which of these options match it correctly? Okay, it's enough time and I will check the chat box and let you know what is the correct answer. Okay. I think there are no answers to this question in the chat box for question number 15. What was the question number 15 again? So the correct answer to this question is option B, 3' CCA and T arm contains pseudouridine and D loop often contains dihydrouridine. This is the correct set of combination regarding the tRNA molecule. Now looking at the structure of the tRNA, this is the acceptor stem, this is the pseudouridine containing C loop, this is the D loop and the anticodon loop. Pseudouridine and dihydrouridine in tRNA. tRNA contains several modified nucleotides that help stabilize its structure and enhance its function. Two of the most important modifications are pseudouridine and dihydrouridine. These modifications play key roles in maintaining the correct folding and function of the tRNA during protein synthesis. Now, what is the function of pseudouridine? Found in the T-Cy-C loop of tRNA, which is the C loop of tRNA. Modification formed when uracil is enzymatically modified by Cy synthase, and its structure is unlike normal uridine. Cy has a CC glycosidic bond instead of an NC bond, making it more stable and flexible. And what is its function? It enhances base stacking interaction, stabilizing the tRNA structure, and aids in ribosome tRNA interactions during translation. Then what is the function of dihydrouridine? Found in the D loop of tRNA, it is formed by enzymatic reduction of uridine adding 2 hydrogen atoms. Unlike standard uridine, dihydrouridine lacks a double bond in the ring, making it more flexible. The dihydrouridine in the D loop increases tRNA stability, ensure proper function during translation, allows flexibility and proper folding, helps in tRNA to adopt its L-shaped 3D structure, and enhance the translational efficiency, improving the interaction with ribosome and enzymes. So is it clear to the students? Very good. So with this, we have come to the end of this problem solving session. I will stop presenting my screen. Yeah. And I hope you guys liked this class and you were able to brush through these concepts once again, even if you were not able to follow the lectures. But I would advise read your lectures, submit your assignments on time, and prepare well for your quizzes and exams. While I am always here and other teaching assistants are always here to clear your doubts, and if you have any questions or suggestions to the style of this class or presentation, you can type in the chat box. All these all these slides will be provided to you via Google Drive, and this is a recorded class, so the recordings will also be uploaded into YouTube, and the links will be shared with you via the NPTEL authority. So with that much of an information, let me end this call. Thank you for joining, and see you next Saturday with problem set 7. Till then, bye bye and enjoy your weekend. I will end this call.